The Science Behind CRISPR by Alec Remai. CRISPR is a revolutionary genome engineering technology. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. In short, CRISPR enables scientists to exploit the immune systems of bacteria to edit genes in other organisms, namely plants and humans. Today it's used primarily for the study of genes and for healing, with the goal to someday even eliminate certain diseases like Alzheimer's and cystic fibrosis through gene therapy. However, together with its great healing potential, there is an ethical dilemma associated with CRISPR as we begin to imagine how it could be used to create genetic advantages and for other non-healing purposes. The story of CRISPR dates back to 1987 when Japanese scientists were studying some E. coli bacteria and they came across an odd set of sequences in the bacteria's DNA. They were palindromic, meaning they read identically forwards and backwards, and they were repeated multiple times in the DNA, being separated only by spacer sequences that were not palindromic. Somewhat confused and intrigued, the scientists wrote, "The biological significance of these sequences is unknown." 3 years later, a scientist by the name of Francisco Mojica discovered very similar DNA sequences in the genetic code of an archaeon. Knowing that archaea and bacteria were of different domains, he concluded that these sequences must be somewhat important to the organism's survival. It was Mojica who gave them the descriptive name CRISPR as an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. After doing some testing using a bioinformatic analysis tool called a Basic Local Alignment Search Tool, or BLAST, he discovered that the DNA found in the CRISPR sequences matched some viral DNA. Although he wondered why a virus's DNA was in a bacterium, the picture became much clearer several years later in 2007. Food scientists were studying the Streptococcus bacteria used to make yogurt and discovered that the CRISPR DNA sequences were part of the immune system. They found that when the bacterium's enzymes managed to kill off an invading virus, smaller enzymes took the remains of the virus's genetic code and cut it up. These cut up pieces of the virus's DNA were then stored in the CRISPR spaces in the bacterium's genome. This immune system strategy is very similar to the science behind the success of vaccinations. The DNA held in the bacterium is like a memory bank. The genetic information there can be used by the bacterium to fend off future similar attacks. However, the way the bacteria fends off similar attacks and viruses it's seen before is very complicated yet effective. Note that the following explanation of the CRISPR immune system will include some information that was discovered later on by various microbiologists. When a bacterium with a CRISPR immune system gets attacked by the same virus a second time, there are two major players in the process of fighting it off. The first is an enzyme known as Cas9. Cas9 is produced when a new viral infection occurs and is important because it can attack DNA by cutting it. Cas9 can make what's called double-stranded breaks, meaning it can cut both strands of a DNA double helix. This cut is achieved through two different domains on the enzyme, Cas9-HNH and Cas9-RuVC. The second major player is a 2-RNA structure known as the guide RNA. The guide RNA itself is made up of 2 RNA strands, CRISPR RNA and tracer RNA. When the viral thread is noticed, and it is established that the virus has attacked in the past, a portion of the DNA from the virus from the first attack, which is held in the CRISPR sequences, is then transcribed into a CRISPR RNA, shortened to cRNA. Each CRISPR RNA consists of a nucleotide repeat and a spacer portion similar to CRISPR DNA in the bacterium. This CRISPR RNA is then base paired to a transactivating CRISPR RNA, shortened to tracer RNA. This tracer RNA will serve as a binding scaffold for Cas9 and as a catalyst in the CRISPR activation process. Once the CRISPR RNA and the tracer RNA are bound together, they are called the guide RNA. So, once the Cas9 enzyme has been produced and the guide RNA has been bound, the two then travel together in the hopes of finding the invading virus at what's known as the target site. When the Cas9 enzyme and the guide RNA arrive at the target site, the Cas9 enzyme will check if the invading virus's DNA matches the genetic information in the guide RNA. If it doesn't, the group will simply move on and keep traveling. But if it does, then the Cas9 enzyme will go to work, cutting up and destroying the invader's DNA. A great comparison is as follows. The Cas9 enzyme is kind of like a hitman. It's always trying to find something to annihilate, but if you don't tell it who to kill, it won't do anything. To aid in this process, the guide RNA is kind of like a mugshot. The Cas9, the hitman, is given the guide RNA, the mugshot, as a reference. In this way, Cas9 can simply compare everything it runs into with the guide RNA, the same way a hitman compares victims to a mugshot. And if there's a match, it can go ahead with the annihilation. It's interesting to note that there is a built-in safety mechanism which ensures that Cas9 doesn't just cut anywhere in the genome of a virus. Short DNA sequences known as PAMs, or protospacer adjacent motifs, serve as tags and sit adjacent to the target DNA sequence. If the Cas9 complex doesn't see a PAM next to its target DNA sequence, it won't cut. All in all, this complicated immune system strategy to defend against returning viruses is very effective. Having understood that CRISPR was an immune system strategy, but not knowing what a cRNA was at the time, a scientist by the name of John van der Oost put himself to work to find any possible use for this knowledge. In 2008, he and his Dutch scientific team successfully transplanted the system to another host bacterium. More notably, they demonstrated that an appropriately designed CRISPR array allowed for specific targeting of any DNA sequence. This meant they could insert artificial CRISPR arrays of viral DNA into a bacterium and as a result make the bacteria immune to a specific virus. This was the first ever case of directly programmable CRISPR-based immunity. However, in 2012, a greater breakthrough occurred as scientists Emmanuel Charpentier and Jennifer Doudna expanded on what Oost had discovered. They discovered the existence of cRNA, one of the components of guide RNA. Their thought process was like this. Since the DNA from Oost's experiments was getting transferred into cRNA and then combined with tracer RNA to form a guide RNA, the scientists hypothesized that it would be possible to simply insert a guide RNA right off the bat. In this way, this single guide RNA could be genetically engineered in vitro and then inserted into a bacterium where a Cas9 enzyme could use it as a mugshot. They also hypothesized that the guide RNA that the Cas9 enzyme uses doesn't have to be virus-specific, thinking that Cas9 will attack anything that matches with its mugshot, be it a virus or otherwise. This was of course shown to be possible in a landmark 2012 paper, and deservingly, Charpentier and Doudna won the 2018 Kavli Prize for Nanoscience. Now that last point that the guide RNA for the Cas9 enzyme doesn't have to be viral DNA is what gives CRISPR its revolutionary status. In theory, any sort of guide RNA can be engineered, given to a Cas9 enzyme, and it can result in the destruction of any DNA we'd like. In a slightly more complicated way, DNA can be inserted as well. This means that we can export this system from bacteria and implement it in crops and humans in the form of a cut and paste gene editing system. For crops, CRISPR can simply be used to enhance our ability to modify plants. Recently, major companies like Monsanto and DuPont have begun licensing CRISPR technology, hoping to develop new crop varieties. Although CRISPR won't entirely replace GMO techniques involving gene transplantation, this gene editing technology will speed up the process and make the edits more precise at the same time. Although gene editing in plants is relatively intriguing, it is far more exciting to think of the possibilities for CRISPR in the domain of human life. And gene editing in humans is a big deal. But before explaining the vast and amazing potential CRISPR gene editing technology has for humans, it is worth noting that there are ethical issues with many of its possible uses. Many people have strong opinions on what is and isn't okay to change in a person's DNA, be it for religious, evolutionary, or other reasons. To show the different types of editing in a way that is easier to interpret, this grid shows two of the most important distinctions when it comes to gene editing: somatic versus germline editing, and therapy versus enhancement. Somatic cells are most of the cells in the body: blood, brain, skin cells, basically anywhere where the DNA doesn't get passed down to the offspring. Germline cells are sperm, egg, or embryos. This is where the DNA does get passed down to the offspring. This is an important distinction because somatic editing results in changes in one person, but germline editing changes future generations and essentially impacts the human population and evolution. In terms of the titles on the side, therapy refers to anything where a disease is treated, whereas enhancements give advantages to people who are already healthy. This is an important line to be drawn as it distinguishes between what someone may need and what someone may want. The top left box is where most of CRISPR technology is focused right now, somatic therapy. This would be any sort of treatment or cure for genetic diseases. Using CRISPR, we could delete the gene causing the disease in the affected person. For example, there are promising cures for cystic fibrosis, sickle cell anemia, Huntington's disease, and Alzheimer's already in development. Other specific diseases, like certain types of cancers where the disease lives in the blood or the bone marrow, are being prospected. For example, blood cancers would be easy to treat with CRISPR because those cells can be removed from the body, edited in vitro, and then replaced back in the body. There's also a potential for treatments for people with type 2 diabetes. We could create skin grafts that can regulate blood glucose levels as insulin does. Diseases like HIV and herpes could also be eradicated. In fact, in 2015, scientists used CRISPR to cut HIV cells out of living cells of patients in a laboratory. More recently, experiments have been conducted on rats which had HIV in 99% of their cells. By injecting CRISPR into the rats' tails, they were able to remove 48% of the virus from the DNA of their body cells. Although these are the uses for CRISPR that will be the most popular in the near future, scientists are still far from ready to declare any disease eradicable using CRISPR. From an ethical point of view, since these are all just cures for diseases for a single person, CRISPR in this case is just being used as a tool for medicine, making it completely ethical. This next box, somatic gene enhancement, is like plastic surgery. It's non-necessary gene editing that stops at a single person. There are no genetic plastic surgeons yet, as the technology is so new. However, it would not be surprising if in the future, one could go see a gene specialist to make them immune to certain diseases. Where the ethical line gets fuzzier is when we enter this quadrant, germline gene therapy, curing future generations. Creating a genetically modified baby is already illegal in over 25 countries, including Canada. In 2015, the UN called for a worldwide moratorium, saying germline modifications could jeopardize the inherent and therefore equal dignity of all human beings and renew eugenics. The fear is that if germline editing were to become available, anyone who'd have access to it would be at least a slight advantage. However, the only people who would have access to it are rich people. Today, kids from wealthier families are already 10 to 15% healthier than kids from poorer families. So the question being asked is whether or not it's socially just for germline editing to even be available. It may result in an even more unbalanced society. Another problem on the right side of the grid is that not everyone agrees on what needs to be cured in future generations. Is deafness a disease? What about Down syndrome or dwarfism? If CRISPR was used to its full potential, those conditions could quite easily be completely eradicated over time. However, most people who have or know someone with one of those conditions likely say, "No, I don't think it should be eradicated." This becomes tricky, as there are many advocate groups who want to preserve their type of being who quite strongly view the prospect of eliminating these conditions as unethical. Already in many Western countries, abortion rates for fetuses with Down syndrome, for example, are extremely high. In the US, it's 67%, France 77%, and Denmark 99%. In response to this, Special Olympian with Down syndrome Frank Stevens asked famously in front of the American Congress, "Is there really no place for us in society?" Back to our grid. If the top right is curing the future generations of diseases, then that makes the bottom right quadrant what we call designer babies. This would be germline genetic enhancement. Like in the movie Gattaca, we could essentially design our baby to have certain traits like gender, eye color, skin color, freckles, and dimples. For complex traits, we might be able to develop a system where we can determine a range of intelligence, appearance, height, and risk of diabetes, for example. This is a weird concept for some people. Would you abort your fetus if you learn that it'll have a low IQ? Are you going to create a genius child with CRISPR? What about looks? For the majority of the world that is religious, it can be difficult to ask these questions. Are we playing God? So the question stands, should CRISPR editing even be allowed? Of course, this is all very far away, and many scientific hurdles must be overcome before anyone starts clinical trials on humans. For example, the Cas9 enzymes can sometimes misfire and edit DNA in unexpected places, which can lead to cancer and new diseases. As geneticist Alan Bradley said, CRISPR's ability to wreak havoc on DNA has been seriously underestimated. There is a lot of scientific research that must occur before CRISPR will be used to its true potential. Despite its complicated ethical future, CRISPR is nonetheless a revolutionary technology. By discovering the amazingly effective immune system of some bacteria, scientists in the last 30 years have changed the future. By exploiting this CRISPR system, we will be able to cure many diseases and stop a lot of suffering. Let's hope that as time marches on, we remain careful with the technology and let CRISPR change the world for the better. Thanks so much for watching.